The overall goal of the following experiment is to non-invasively monitor real-time metabolic alterations at the whole animal level within glycolysis, pyruvate metabolism, and the tricarboxylic acid cycle that are caused by individual metabolic pathway genetic alterations and/or pharmacologic treatments. This is achieved by feeding stable isotopes to living nematodes to provide sufficient isotopic enrichment in worm metabolites to permit quantitation of genetic and/or pharmacologic alterations in intermediary metabolic pathways. As a second step, isotopic incorporation in atmospheric and dissolved carbon dioxide are measured by gas ratio mass spectrometry, which provides more direct assessment of intermediary metabolic flux over short periods of time. Next, samples are processed for whole worm-free amino acid analysis using HPLC, as well as by isotopic enrichment in amino acids and organic acids by GCMS, in order to quantify metabolic flux alteration in nematodes following stable isotope exposure. Results are obtained that show effects of variable durations of isotope exposure, bacterial clearing, and alternative worm disruption in wild-type worms to interrogate isotopic abundance in intermediary metabolites.
JoVE's method can provide insight into C. elegans intermediary metabolism. It can also be applied to other systems, such as mammalian cell lines and tissues.
We first had the idea for this method when we observed consistent abnormalities in free worm amino acid analysis by HPLC that were suggestive of impaired intermediary metabolic flux in primary respiratory chain dysfunction.
To begin this procedure, worms that have been exposed to a stable isotope